Hello.
Hello.
Welcome to my very procrastinated presentation titled Lymphatic Remodeling in Cancer. So I know this week's topic was supposed to be angiogenesis in cancer, but I was like, wow, blood's great, but it's really basic. And I really like studying the lymphatic system because it is super underrated. So instead of doing angiogenesis, we're going to be looking at lymphangiogenesis and its role in like metastasis with tumors. So just to kind of clarify, lymphangiogenesis is just like a fancy term that we use to talk about like the development of lymphatic vasculature. Cool. And if at any point you guys have questions, just like stop me, obviously. All right. Dang it, I have to click it. Oh, okay, there. Okay, nice. So we're gonna start off by talking about two contrasting models which suggest the dissemination of metastatic tumors to distant organs. And this is kind of like the debate of like, is it blood? Is it lymph? What's more important? And obviously lymph isn't as widely studied. But when we look at the two different models, the first one talks about cancer metastasis occurring independent of the lymphatic system. And it talks about how blood vessels are responsible for invading tumors, carrying the tumors to different locations. And that in this specific model that they've proposed, the lymphatic system doesn't necessarily play an actual role in the metastasis. Instead, they say it's just a reflection of how aggressive the tumor is. Whereas the lymphatic dependent sequential model, which is what I'm going to be kind of talking about today, proposes this alternative model that suggests that tumor cells spread via the lymphatic system and lymph nodes, so that there's an actual role that it has. So that's kind of what we're going to be looking at today. All right, so before we jump in officially, I'm just going to kind of preface with the significance of lymphangiogenesis in tumor metastasis and kind of like the big question of why is this important and why do we care? So obviously majority of cancer deaths are attributed to metastatic cases, and obviously those tumors have to travel through a network either in blood or what we're going to talk about today, which is through your lymph. So lymphangiogenesis, or again, that development of the lymphatic vasculature, as well as lymphatic enlargement, can facilitate the metastasis of these tumor cells. And the actual presence of tumor cells in SLNs, which are your centennial lymph nodes, is an indicator of poor outcomes in cancer. So being able to study lymphatics, either in terms of their proliferation or in their enlargement, can be an indicator that we can use in prognosis. And then essentially putting all this together, understanding the signaling pathways can provide us with insight on how we can develop prognosis as well as therapeutic strategies. Okay, cool. Just like Arvin's presentation last week, we have a set of objectives, woo! So the first of which is to understand the development and structure of a lymphatic system. And structure relates to function, so I'm talking about function relative to that. The central signaling axis that we're going to be looking at today is going to be the VEGF-C and VEGF-D signaling axis. So those are the ligands, and it's going to basically work with this receptor called VEGFR3. So this specific signaling axis is what induces lymphangiogenesis with tumor metastasis. And we'll be looking at it in the context of in both primary tumor remodeling as well as remodeling outside of that in the collecting lymphatics. And then finally, understanding how we can apply the signaling axis and kind of how it functions relative to tumor metastasis in cancer prognosis and therapeutics. So we're going to start with a crash course in the lymphatic system. So obviously kind of what we've learned is that lymphatic vasculature plays an essential role in tissue fluid homeostasis, in immune functioning, as well as the absorption of dietary fat. And just to kind of go over a brief overview of its structure, it is a network that is comprised of these blind-ended vessels. So you can kind of see here, blind-ended vessels, the sacs, and you can see how interstitial fluid as well as cells can enter through these permeable layers, and they travel unidirectionally through the different types of lymphatic vessels that we have. So the three that we have are the initial, so the capillaries. So you kind of see that diagram where it's like the network of like oxygenated blood in red, the oxygenated blood in blue, and then that green thing going through, yeah, that green thing, those are your lymphatic capillaries, and those are your initial vessels that we have. And those are the ones that are kind of indicated here that are highly permeable. And these are the ones that actually proliferate when we talk about lymphangiogenesis, so the sprouting and the budding of those specific vessels. The next set, so we kind of go from the initial through the pre-collecting lymphatic into this classification, which is the collecting lymphatic. So these are larger, and essentially you have the initial lymphatics draining into the collecting, and they're divided into these sections by these valves, which again will essentially allow the flow of lymphatics or lymph, to go in that one direction. And then finally, kind of like the third one are the ducts. So they're large, they connect to your subclavian veins, and they return the lymph to your blood circulation. So that's just kind of like the general flow of lymphatics. Interesting thing is that when we talk about cancer patients, lymphatic vessels can actually be influenced by tumor-derived growth factors, which is what we're going to be talking about today. And these said growth factors can prompt dynamic changes and remodeling in both like the proliferation, so the amount of lymphangiogenesis and like the growth of those vessels, as well as the enlargement of the actual circumference of those vessels. And that is what will attribute that metastatic spread to both the lymph nodes as well as the distant organs. Okay, so kind of elaborating a little bit on, we have an understanding of the basic structure, but we're going to talk about how the lymphatic vessels themselves go through the process of development, and how certain markers and indicators and factors that attribute the development and specification of lymphatic vessels are key to understanding the signaling axis. So the word angiogenesis, like I mentioned earlier, is the term that we give to the development of new blood vessels. And so lymph angiogenesis, like I mentioned, is going to be the development of those lymphatic vessels. And the interesting thing is that the lymphatic vessels actually develop secondary, so after the development of those blood vessels, but in a very parallel process. So that's kind of why we see a lot of studies being conducted. We have to study like lymphatics relative or like parallel to the blood, is because they have a very like inner related process. So Florence Sabin is the name of a scientist who developed the currently most widely accepted model. And he kind of talked about the development of these vessels in a four-step process, which is lymphatic competence, lymphatic bias, lymphatic specification, and LEC differentiation. Just really briefly, LEC stands for lymphatic endothelial cells. So those are kind of like the primary cells that we have when we talk about like lymphatic vessels themselves. So when we kind of talk about the context of going from blood vessel to lymphatic vessel, in embryonic development, we have blood vessels that originate from these mesodermally derived endothelial cell precursors. So vessels will then grow and remodel and sprout and split, and this forms the blood vessels or through angiogenesis. And then lymphatic vasculature then appears after the blood vasculature forms. So in that first step of lymphatic competence, these kind of like these precursor cells are those venous endothelial cells. They're competent, or they are then able to essentially be receptive to lymphatic inducing signals. And there are certain gene products that are needed to develop these lymphatic vessels, in addition to the factors that are needed for angiogenesis. So one of them, if you can see from here in this part one, is LIVE1, which stands for lymphatic vessel endothelial polyuran receptor 1, which is a mouthful, but LIVE1 is essentially that first indicator that shows that you are developing a lymphatic vessel as opposed to a blood vessel. And the interesting thing, as you can see from the image, is that LIVE1 by the color here is uniformly expressed, which is different from what we see in the second part, which is lymphatic bias. So in that second step, preceding the expression of LIVE1, there's this induction of the expression of this other factor called PROX1, which stands for Prospero-related homeobox 1. But essentially, it's this other growth factor that is expressed in polarity, and therefore you actually see the budding of the lymphatic vessels occurring on one side as a result of the expression of PROX1. And this is kind of the point at which the lymphatic vessels, so these venous endothelial cells, then start to take on the lymphatic cell phenotype. We go into lymphatic specification. So we kind of see here this increase in several different factors, and VEGFR3 is one of them. This is expressed in all LECs once they've been specialized into that specific cell. And then kind of moving forward, you get the differentiation of LECs, or your lymphatic endothelial cells. Sorry. And those like three different kinds of talk about, so the initial, the collecting, and then the actual like ducts. So the important kind of like character I want to point out here, or the factor, is VEGFR3. And that's going to be basically a marker that tells you that this is like a lymphatic endothelial cell. Oops. Okay.
Oh,
So,
sorry, I have a really quick question. Sorry to interrupt.
No, you're good.
Can you go back to the previous slide really quick? Maybe I missed this, but so based on the coloring here, is that indicating the lymph derives from, I guess, a precursor phenotype that's somewhere between arteries and veins, like how this image is part blue, part red?
So that's a great question. I'm not entirely sure in terms of the arteries and veins here. In terms of the color, I should check on that.
Okay, so
all I know for sure is that, you know what, that's a good question. I'll check on that. I should have considered that. All I know is that the importance of what I was trying to get to was that this specific factor, PROX1, is expressed on one side, which is why you kind of see the budding coming off of one end as opposed to the other. And that's why you kind of see the growth. You talk about like lymphatic or lymphangiogenesis, it'll bud off at a certain point. And it only stems from where the expression of that PROX1 is coming from.
Gotcha. Okay, thanks.
But I'll check. That's a good question.
Does that have something to do with that, like E10.5, E something or other that's on there? If you happen to know what that is, or I don't know if I know that's related.
Wait, where is it?
I think I drew over it.
Like B.
So,
like,
I think this is basically just referring to like the embryo, like the embryonic day, because at different points in time over development, you'll see the expression of these different like growth factors, and it kind of happens in sequence. So if you can kind of see like from 0 to 9.5, you just kind of get an initial expression, and then over time, like shortly after, you see like a day and a half later, whatever, you can see the expression of another one, and then over time, when you get to like 14.5, that's when you have the functioning lymph. So over the times that they're an embryo to developing, that's all that the E represents, if that makes sense. Sorry. Okay, cool. All right. Wait, Tim, how do you get rid of the blue thing on like?
Let me figure that out.
All right, you're good. That's fine. Where were we? Okay, cool. There's a green thing now. Okay. So, like I mentioned, the kind of big thing that you want to take away from the last one is that you have blood vessels, and then there are these specific growth factors that you develop that are expressed to ensure that you get lymphatic vessels as opposed to blood vessels. And one of the main markers is this thing called VEGFR3, which is vascular endothelial growth factor receptor. So when you have that present in your cell, that basically means, yes, this is a lymphatic vessel cell, not a blood vessel cell. So the important thing to note here is that LECs, so the lymphatic endothelial cells, and BECs, blood endothelial cells, do share a lot of common cellular and molecular traits, but obviously they are different and unique in their own respective molecular and physiological behavior, and they have their own respective signaling pathways that allow them to develop and function as such. So in the context of like cancer research and lymphatic studies, the most widely studied like signaling pathway with respect to like tumors and lymphangiogenesis is this one that is called the VEGF-C or VEGF-D signaling axis with VEGFR3. So a lot of acronyms, but just to kind of like simplify and clarify it, The VEGF-C and VEGF-D, those are two different but kind of similar ligands, and then your VEGFR3 is the receptor. So the interaction between that ligand, those ligands, and then that specific receptor is essentially what will lead to lymphangiogenic growth. And then the other important thing to note is that VEGF-C and VEGF-D, those two ligands, are secreted by tumors. So if you can kind of see from the diagram here, you have VEGF-C and VEGF-D, which is the ligand, fitting into VEGFR3 here, which is this like transmembrane receptor. And this is like nitty gritty molecular stuff, but it kind of activates this like downstream signaling pathway, which ultimately leads to lymphangiogenesis. So you have survival of LECs, migration, as well as proliferation of those lymphatic endothelial cells. Next one. Okay. Whoops. Sorry, I have like two computers going. All right. So overexpression of VEGF-C, that specific ligand, in cancer cells can therefore induce that signaling axis, so that interaction between ligand and receptor, and that in turn will induce both intratumoral as well as peritumoral lymphangiogenesis. So intra obviously means like within the tumor, peri is around. And then the interesting thing is that we have kind of found differences in the presentation of if it's intratumoral versus if it's peritumoral. So when we talk about intratumoral lymphangiogenesis, it hasn't necessarily been found to be essential for increasing the metastasis of the tumors to the lymph nodes. So like the tumors won't necessarily need to be like within the tumor in order for it to occur. But then what they observed with the peritumoral lymphatics, so the lymphatics that grew around the tumors, is that they observed hyperplasia, so a lot of uncontrolled growth of LECs, and then by having this like really intense proliferation of these lymphatic endothelial cells, you're increasing the surface area of the lymphatic vessels in contact with the tumor, and that in itself, because of those LECs at the junctions being very permeable, it's possible there then for the tumors themselves to enter and then kind of use the lymphatic system then as a route to travel, and either, you know, the tumors will enter like the lymph nodes, or it can use that same like network throughout the body to travel to like a distant organ. Where's the click? Okay, cool. So a lot of studies were conducted essentially that looked at the remodeling of these lymphatic vessels at the primary tumor level by essentially studying this axis between VEGF-C, VEGF-D, but in this case, we're going to talk about VEGF-C and VEGFR3 again, so ligand and receptor. So one study, essentially, they looked at VEGF-C, and then they took a soluble form of the receptor VEGFR3. So it basically meant that they were taking off some of those binding domains so that the ligand and receptor interaction wasn't happening the way that it's actually supposed to. And they found that that was successful in restricting lymphangiogenesis in the tumors and around the tumors. And they saw a reduction in lymph node metastasis. So that interaction between VEGF-C and VEGFR3 is really crucial in the development of the lymphatic vasculature in terms of cancer. They did a similar thing where they wanted to block the ability of the binding, but not through the soluble receptor, but they used those monoclonal antibodies that were specifically targeted for VEGFR3. And again, by blocking that interaction for the receptor ligand kind of axis, you also saw the same thing with restricted lymphangiogenesis. And apart from just looking at that direct interaction, they've also looked at it in the context of things that were more upstream, so things that could affect the ability of VEGF-C or VEGF-D to be produced. So WNT1 or WNT1 is the factor that will essentially, whoa, where is it? Suppress the expression of VEGF-C. So when they implemented that, expressed it, and then it kind of reduced it, therefore VEGF-C wasn't really present in order to bind with its receptor. They saw delayed lymphangiogenesis, which is an example of a therapeutic target they can look at. And then the other one that they looked at was the role of prostaglandins in increasing the expression of VEGF-C. So they found that when they had prostaglandins and it induced the expression of VEGF-C the, again, receptor-ligand interaction. It promoted lymphangiogenesis. And then a few other important molecules that they also studied was the interaction of COX-2, which was cyclooxygenase 2. And essentially that COX-2 molecule is responsible for synthesizing prostaglandins. So when you had an increased expression of COX-2, you increased amount of prostaglandins, and then its ability for those prostaglandins to bind to their respective receptors. That's where you also saw the pathway to the receptor ligand interaction with VEGF-C, VEGFR3. And then you again got the promotion of lymphangiogenesis. And then the other enzyme that they looked at was 15-PGDH, or hydroprostaglandin dehydrogenase. And instead of increasing the amount of prostaglandins being synthesized, this specific molecule is responsible for breaking down prostaglandins in a way that makes them be able to be exposed to LECs for a longer period of time. So apart from just having an increased amount of the prostaglandins present, by actually making them in a form that could be exposed to the LECs for a prolonged period of time, that also resulted in vessel enlargement, so remodeling of those lymphatic vessels. Okay, so another thing I do want to point out is that outside of just that interaction of the signaling axis is that the lymphatic system itself, so the LECs along here, there's also an important role that the extracellular matrix, or the ECM, plays in supporting it. So because of the extracellular matrix kind of working in tandem with it, you're going to need that specific structure in order to ensure that there's enough support and that the interactions occurring between these different signaling axes are being modulated. So the important actor in this case is this complex of A4 and beta 1. So in this complex here, which is essentially responsible for mediating the interaction between your LECs, or your lymphatic vessel cells, and the extracellular matrix, is that when one basically genetically removed it, so lost the gene to create this complex, there was no tumor-induced lymphangiogenesis. And then two, when they implemented antagonists such as this, it also suppressed the lymphangiogenesis occurring with respect to the tumors. So apart from just that direct signaling axis between VEGF-C, VEGF-D, and VEGFR3, by basically interfering with this complex that also ensures the interaction between the two different systems, so the supporting system and the lymphatic system, that also has a role in reducing metastasis. And then the other kind of part of the paper that was really interesting was how, apart from just the signaling axis kind of operating its functionality, is the question of how does, how do the molecules themselves kind of concentrate in this area? And it was the role of chemokines and how they are able to promote the ability of these cancer cells to kind of find their way to the LECs themselves. So if we kind of think of it in terms of a metaphor, you can think of the lymphatic system itself as a highway, and you have several cells that are traveling along it. One of them, or one classification of them, being antigen presenting cells, or APCs, that kind of act as the vehicles that travel along them. And then these other molecules that are your chemokines kind of work as your navigators, or your Google Maps or whatever. So what's interesting is that this system can actually be exploited in a way that will bring or navigate those cancer cells to your lymphatic cells. And in that way, it's going to kind of allow the cancer to make its way to the lymph and travel through it, and then travel to either the lymph nodes or to distant organs. So what this image is basically pointing out is this thing over here, the receptor CCR7. I'm just going to also simplify this when I say there's a lot of molecules of names and acronyms, but if you see an R, that's receptor. You can kind of see it by the little thing here, and then L is ligand. So CCR7 is on the tumor cells, so cancer cells labeled here. And it acts as a receptor for these ligands, CCL21, as well as another one that was mentioned in the paper, CCL19. And then once again, we talk about the signaling axis, and the role of VEGF-C and VEGF-D is that the tumor cells, like we talked about in the beginning, will secrete these ligands. So VEGF-C, VEGF-D, and a few others. And then what happens is that, again, VEGF-C, for example, will bind to its receptor, VEGFR3, on the LEC. And then as a result of that binding, the LEC will increase the secretion of the ligand of CCL21. As you can kind of see, there's an increased concentration here. And then these cancer cells that have the receptors, so they're positive for that a receptor that binds the ligand, will therefore be attracted to the LECs. So that was also a significant feature that they had found in their studies. And then the other part of this was, okay, cool, we have determined that the cancer cells are able to approach the LECs, but how are they able to enter and use that lymphatic system as the actual highway to travel the system of the body? And they looked at this other set of receptor ligand interactions, specifically through CCR8 and its ligand CCL1, and they essentially determined that this is a potential checkpoint receptor ligand interaction that would allow the entry of tumors into the lymph nodes. So the paper suggested that there were studies that found that by blocking that CCR8, the receptor itself, by inhibiting it, you can actually restrict tumor metastasis because it's not able to enter the LEC and therefore travel the lymphatic system. Okay. So now that we've kind of talked a little bit about the primary tumor, it's important to note that beyond just the remodeling of lymphatics around that initial primary tumor, there are other ways that lymphatics can remodel. So we're going to be talking about three. So the first of which is basically how VEGF-C, we talked about how in the previous set of examples we discussed, how the signaling axis can allow this proliferation of LECs, which causes lymphangiogenesis. So it's more vessels. But when we talk about the secondary type of our lymphatic vessels, which are collecting ducts, so not the capillaries, not the initial ones, but the collecting ducts where they drain, the role here is that prostaglandins can actually mediate this expression of VEGF-C, and then in these larger collecting ducts, they can be enlarged even more. And that is also going to kind of make it easier for those tumor cells, once they've kind of entered, to be able to pass and transport. The second way in which remodeling can occur is that we encounter what's called lymphascular niche. So lymphascular niche is basically a term that is used to refer to the expansion of the lymphatic network in the draining lymph nodes. So within the lymph nodes themselves, there is an increase in the density of lymphatic vessels present, and that in itself will also facilitate an increase in the transport of those tumor cells. And like we talked about in the beginning, when we kind of analyze patients or we look for signs that there's invasive cancer present, you look at the SLNs, so those centennial lymph nodes, and if you see the presence of the tumor cells there, that's pretty indicative that is pretty aggressive and advanced cancer. There's a significant amount of tumor progression that has occurred. So when we look at that lymphascular niche and the development of those multiple vessels in the nodes, that kind of does indicate this potential that it's using that transport as a way to metastasize. And then the other interesting feature that the paper talked about outside of primary tumor remodeling is the fact that LECs share a lot of different characteristics or similar characteristics with those antigen presenting cells, and in a way that actually contributes to there being a degree of tumor immunity with these cells. So kind of going back to basic microbiology, your cells can express your histocompatibility complex molecules, class 1 or class 2. Class 1 is basically used as a self-marker in all nucleated cells, and then the class 2 is what's used for antigen presenting cells. So in the case of these LECs, they express both the self marker as well as the APC marker. And then as a result, that kind of allows for them to have like a special categorization of like being an APC. So when we talk about VEGF-C, that ligand again being associated with these LECs, VEGF-C has this weird characteristic that they found that it's actually able to protect melanoma cells from anti-tumoral immunity. So it's not recognizing it as like a foreign invader per se. And there, like other studies have also found that LECs cross-presented tumor-associated antigen and promoted the apoptosis of tumor-specific CD8 positive cells. So in essence, that basically means apart from evading the immune system in terms of saying, I'm not a foreign invader, they're also able to destroy the components that are supposed to protect, you know, the immune system from like foreign invaders. So it hides and it destroys the like things that are supposed to keep the body safe. Okay. So we are kind of getting toward the end here. And one thing that the paper talked about is kind of like the applications of why it's important to understand lymphangiogenesis in the context of like tumor progression and metastasis. And what they found was that looking at the development of lymphatic vessels or lymphangiogenesis, it's a way that we can study or potentially look at the indications of cancer outcomes. So a few examples I provided. One is that when they studied patients with melanoma, was that they found that with the metastatic melanomas, there was an increased incidence of intratumoral lymphatics, as well as like enlarged tumor-associated lymphatics. And when they identified those specific cases where the lymphatics had been remodeled in these patients, their tumors, there was a higher percentage of them who had poor disease-free and overall survival. So not only were these patients with melanoma exhibiting, like who exhibited remodeling of their lymphatics, surviving less in terms of like getting rid of the cancer, but overall they had poor outcomes. So like they typically probably didn't survive melanoma. Another example that they looked at was in breast cancer. So when they looked at inflammatory breast cancer, because obviously we talk about the lymphatic system and its regulation of interstitial fluid. So inflammation is of course an abnormality and a component of that. There was a proliferation of the LECs and lymphangiogenesis anytime that they saw like really severe cases of inflammatory breast cancer. And this is potentially, it's been proposed as a reason why breast cancer itself is like very aggressive and able to spread through the lymphatic system. And then the last kind of example that they really went into was gastric cancer. So what was interesting was that they were able to identify that patients who had elevated levels of that VEGF-C ligand often had poor prognoses when they compared them to those patients who had gastric cancer but had low expression of the protein. So that is also an indication that that specific receptor ligand interaction that is responsible for lymphangiogenesis and lymphatic enlargement plays a significant role in tumor metastasis. So essentially, what does this mean for like translational medicine and how can we apply this to developing therapies? And the main things that we talked about today were the VEGF-C and VEGF-D interactions with the VEGFR3 receptors. So a lot of the therapies being developed right now with respect to lymphatics and cancer are targeted at inhibiting this interaction along this axis. So we talked about this a little bit earlier, where they can implement these neutralizing monoclonal antibodies that will target either the ligands VEGF-C or VEGF-D, as well as the receptor VEGFR3 or NRP2, which is a co-receptor of the VEGFR3 receptor. Again, we talked about soluble VEGFR3, which will essentially prevent the binding domains and the functionality of that signaling axis. And then a few others that have been proposed are protein kinase inhibitors, these angioproteins that are being targeted by antibodies as well as the non-steroid anti-inflammatory drugs that can target COX. And once again, COX were the ones that were responsible for enlarging the secondary or the drain, like the collecting lymphatic vessels themselves. So these are all techniques that are being implemented to date. And yeah, the monoclonal antibodies apparently entered phase 1 clinical trials in 2013. So progress being made. And just to kind of conclude, to talk about where the future of this field of study can go. Even though they have identified some promising leads in terms of understanding the signaling axis itself, there's still a lot of things that remain unanswered, obviously. The first of which is that they're still not 100% sure of the molecular mechanisms that allow the entry of tumor cells into the lymphatics. So we did talk about the CCL1, CCR8 interaction, but that still hasn't been very widely studied if there's other factors involved in it. Another one is they have to determine whether or not tumor lymphangiogenesis and remodeling are absolutely required for metastasis to lymph nodes or distant organs. So it's still the question of, is it blood, is it lymph? We're still trying to figure out, especially because the two, as we saw, are developing in a process that's consecutive, and how those factors can interact and influence each other. So that still has to be determined. And then the third one is to define the degree to which lymph node metastasis contributes to the spread of cancer to distant organs. So in a lot of cases, like we talked about with the melanoma, the gastric cancer, and the breast cancer, it is true that they've identified a lot of evidence in terms of saying, well, if we see it in the lymph nodes, it's a pretty significant indicator that it's able to spread, but is that always the case? Until now, it hasn't been determined. But at the end of the day, I think understanding the role that the lymphatic network plays and the development of these lymphatic vessels is significant to how we study and screen and treat cancer. So right now, a lot of clinical or translational applications will screen the blood for these proteins, nucleic acids, exosomes, all these things, because we've thus until now basically said, oh, it always goes through the blood. So we'll probably find evidence of the metastatic tumors in the blood. But now that there's a growing body of evidence that suggests that the lymphatic system does play an important role, it kind of introduces the idea of, well, why don't we start screening lymph nodes for some of those similar things? So yeah, that concludes my presentation.